Hi, I'm Ben Romanjenko, and I'm working with Dr. Wyagno of the URSP lab in the Department of Botany. I'm looking for chloroplast-related genes in a brassica transcript dataset containing control and mild drought plants. Brassica was used in the initial study for its wide agricultural application seen by the variety of plants above, a quick growth cycle, high reproductive rate, and a mature reference genome. Additionally, it has a developed chloroplast genome that will support my work. Intentions of the initial study looked at phenotypic and transcriptomic responses with hopes of building the background for others to breed plants that produce better yields even in adverse environments. The measurements used to best study the environmental stresses are related to traits and functions that occur in the chloroplast. Understanding drought's effects on a plant requires some background regarding the processes that occur in the chloroplast. First, the light reactions occur in the thylakoid membrane, where light harvesting complexes and reaction centers contain electrons that are excited by photons. This energy follows three competing paths: photosynthesis, heat, and fluorescence, such that as when environmental conditions become less favorable, photosynthesis decreases and heat and fluorescence increase. Second, thylakoid lumen is where water splitting occurs. This is to resupply the electrons for photosynthesis. Third, the stroma, where the Calvin cycle occurs. CO2 is fixed and converted to sugars. Measurements of CO2 entering and being used by the plant versus respiration, where CO2 is produced, can be used to determine the photosynthetic rate of chloroplast, cell, and plant. In normal conditions, these measurements are often cyclic in nature. Drought is known to decrease water splitting and gas exchange while increasing fluorescence. The previous analysis compared transcript levels between time of day and control versus treatment. In the graphic, you can see the strong relationship between time of day and daily variations in gas exchange, fluorescence, and sugar accumulation. Observing both transcript levels and physiological measures being strongly associated with time of day, it is expected that genes associated with chloroplasts and photosystem II will have a similar or greater correlation between treatment due to the number of chloroplast functions being altered, which poses us to ask the question: how do transcript levels associated with chloroplast and photosystem II respond to drought treatment along a time course? In this, I'll develop a pipeline that uses gene ontology information to pull out chloroplast 2 and chloroplast and photosystem II genes from the previous data set, and then rerunning differential pattern analysis via linear models, or DPALM, and weighted correlation network analysis, WGCNA, to understand the relationship of transcripts to time course or treatment. Depending on the level of success for identifying chloroplast-related genes, it may be necessary to realign the transcripts to a chloroplast reference genome via ChloroSeq. We will then use DPALM and WGCNA to identify relations between chloroplast transcript levels, treatment, and time course. Possible outcomes include low chloroplast transcript levels, possibly due to improper harvesting or sample prep. Otherwise, a sufficient number of transcripts will be identified and built into an expression network. Knowledge gained from either result will help inform upcoming experiments.